Hello, and welcome to Calm Science Nights. I'm so glad you're here with me tonight. Before we begin our peaceful journey through the science of aging, if you find comfort in drifting off to the soothing rhythm of biological mysteries, please consider subscribing to the channel. It helps us create more of these tranquil explorations together. Now, settle into your most comfortable position. Let the weight of the day lift from your shoulders. Tonight, we're going to explore one of life's most universal and inevitable processes, aging. Why do we grow old? Why does our hair turn gray, our skin wrinkle, our bodies gradually lose the vitality of youth? Over the next 100 facts, we'll trace humanity's journey from ancient observations about mortality to modern molecular biology, revealing the intricate mechanisms that cause us to age. There's no need to remember everything tonight. In fact, there's no need to stay awake at all. Let these words wash over you like gentle waves, each fact a soft ripple in the stream of understanding, carrying you peacefully towards sleep. Just allow the story to unfold at its own unhurried pace, the way time itself passes, steadily, inevitably, marking the passage of our lives. Close your eyes now, and feel the quiet rhythm of your breathing, the steady beating of your heart. These processes have continued since the moment you were born, and they will continue throughout your life. Your body is a remarkable biological machine, constantly renewing itself, repairing damage, maintaining the delicate balance that keeps you alive. And yet, despite all this maintenance, despite the body's best efforts, time leaves its mark. We age. This is not a failure but a fundamental feature of being alive. Our story begins not with modern science but with the simple observations humans have made throughout history. People have always noticed that the very young and the very old look different from those in the prime of life. Babies are smooth-skinned and helpless, gradually growing stronger and more capable. Adults reach a peak of physical ability, a time when the body seems most vigorous and resilient. But then, sometime in middle age, the tide begins to turn. Recovery from injury takes longer. Hair loses its color. Skin becomes less elastic. Energy wanes. This pattern is so universal, so obvious, that every culture has recognized it. Ancient philosophers tried to understand why this happens. The Greek physician Hippocrates, often called the father of medicine, lived in the 5th century BCE and believed that aging was caused by the loss of body heat. He observed that young people are warm and vital, while old people are cold and frail. He thought that life was a process of slowly cooling down, like a fire burning lower until it finally goes out. This was a poetic idea, an attempt to find a unifying principle, but it wasn't quite right. The body doesn't simply cool, the process is far more complex. Another Greek philosopher, Aristotle, who lived in the 4th century BCE, proposed that aging was caused by the gradual loss of moisture in the body. He noticed that young skin is plump and moist while old skin is dry and wrinkled. He thought that life was a process of drying out, that we are born wet and gradually desiccate over time until we become too dry to sustain life. Again, there was an element of observation here. Skin does lose moisture with age, but the explanation was incomplete. Aging involves much more than simply drying out. These ancient theories, while incorrect in their specifics, were asking the right question: what is the fundamental process underlying aging? For centuries, this question remained unanswered. Aging was simply accepted as natural, inevitable, mysterious. People noticed that different species aged at different rates. A mouse might live only 2 or 3 years, while a human could live to 70 or beyond. Some trees lived for centuries. But no one understood why these differences existed. The scientific study of aging, what we now call gerontology, really began in the 19th and early 20th centuries as biologists started to understand the cellular basis of life. The cell theory, established in the 1800s, revealed that all living things are made of cells, and that these cells come from the division of existing cells. This raised an intriguing question. If our cells can divide and create new cells, why can't our bodies renew themselves indefinitely? Why do we age at all if our cells can be replaced? One of the first major insights came from August Weismann, a German biologist working in the late 1800s. Weismann made a crucial distinction between two types of cells in the body: germ cells, the cells that become eggs and sperm, and somatic cells, all the other cells that make up the body. Weismann proposed that germ cells are potentially immortal. They can be passed from generation to generation indefinitely. But somatic cells, he suggested, are mortal. They have a limited lifespan because, from an evolutionary perspective, they don't need to last forever. Once an organism has reproduced and passed its genes to the next generation, there's no evolutionary advantage to maintaining the soma, the body, indefinitely. This was a profound insight. Aging might not be a design flaw but rather a consequence of how evolution works. Natural selection favors traits that help organisms survive long enough to reproduce and raise offspring. But once reproduction is complete, the force of natural selection weakens. There's little evolutionary pressure to maintain perfect cellular repair mechanisms for individuals who have already passed on their genes. This idea, that aging is related to the declining force of natural selection with age, would become a cornerstone of evolutionary theories of aging. Weismann's ideas were theoretical, but they pointed scientists in a productive direction. If aging is programmed into our somatic cells, then perhaps we could understand it by studying what happens to cells over time. This line of thinking led to one of the most important discoveries in the biology of aging: the finding that normal human cells can't divide forever. In the early 1960s, a biologist named Leonard Hayflick was studying human cells grown in laboratory culture. Previous scientists had believed that cells, given the right nutrients and conditions, could divide indefinitely. But Hayflick noticed something different. When he took normal human cells and grew them in culture dishes, they would divide enthusiastically at first, roughly doubling in number with each division. But after about 40 to 60 divisions, something strange happened. The cells would slow down and eventually stop dividing altogether, entering a state called senescence, a kind of permanent retirement where the cells remained alive but could no longer reproduce. This discovery, which became known as the Hayflick limit, was revolutionary. It showed that normal human cells have a built-in counting mechanism, some kind of cellular clock that keeps track of how many times they've divided. After reaching their limit, they simply stop, as if they've run out of divisions. This was direct evidence that aging happens at the cellular level, not just at the level of the whole organism. Our cells themselves grow old. And so our journey has begun with the simple observations humans made throughout history. People always noticed very young and very old look different from those in life's prime. Babies smooth-skinned and helpless, gradually growing stronger and more capable. Adults reach peak physical ability, time when body seems most vigorous and resilient. But then sometime in middle age, tide begins to turn, recovery from injury takes longer, hair loses color, skin becomes less elastic, energy wanes. Pattern so universal, so obvious that every culture recognized it. We learned ancient philosophers tried understanding why this happens. Greek physician Hippocrates, often called father of medicine, lived 5th century BCE, believed aging caused by loss of body heat. Observed young people warm and vital while old people cold and frail, thought life was process of slowly cooling down like fire burning lower until finally goes out. Poetic idea attempting to find unifying principle but not quite right. Body doesn't simply cool, process far more complex. We discovered another Greek philosopher, Aristotle, lived 4th century BCE, proposed aging caused by gradual body moisture loss, noticed young skin plump and moist while old skin dry and wrinkled, thought life was process of drying out, born wet, gradually desiccate over time until too dry to sustain life. Again, element of observation, skin does lose moisture with age, but explanation incomplete. Aging involves much more than simply drying out. We found these ancient theories, while incorrect in specifics, were asking right question. What is fundamental process underlying aging? For centuries, question remained unanswered, aging simply accepted as natural, inevitable, mysterious. People noticed different species aged at different rates. Mouse might live only 2 or 3 years while human could live to 70 or beyond. Some trees lived for centuries, but no one understood why these differences existed. We learned scientific aging study, what we now call gerontology, really began in 19th and early 20th centuries as biologists started understanding life's cellular basis. Cell theory, established in 1800s, revealed all living things made of cells, and these cells come from existing cell division. Raised intriguing question: if our cells can divide and create new cells, why can't our bodies renew themselves indefinitely? Why do we age at all if our cells can be replaced? We discovered one of first major insights came from August Weismann, German biologist working late 1800s. Weismann made crucial distinction between two body cell types: germ cells, cells becoming eggs and sperm, and somatic cells, all other cells making up body. Weismann proposed germ cells, potentially immortal, can be passed generation to generation indefinitely, but somatic cells are mortal, have limited lifespan because from evolutionary perspective don't need to last forever. Once organism reproduced and passed genes to next generation, no evolutionary advantage to maintaining soma, the body, indefinitely. We found this profound insight. Aging might not be design flaw but consequence of how evolution works. Natural selection favors traits helping organisms survive long enough to reproduce and raise offspring. But once reproduction complete, natural selection force weakens. Little evolutionary pressure to maintain perfect cellular repair mechanisms for individuals already passed on genes. Idea that aging related to declining natural selection force with age would become cornerstone of evolutionary aging theories. We learned Weismann's ideas were theoretical but pointed scientists in productive direction. If aging programmed into somatic cells perhaps could understand by studying what happens to cells over time. This thinking line led to one of aging biology's most important discoveries: finding that normal human cells can't divide forever. We discovered in early 1960s biologist Leonard Hayflick studying human cells grown in laboratory culture, Previous scientists believed cells given right nutrients and conditions could divide indefinitely. But Hayflick noticed something different. When took normal human cells and grew them in culture dishes would divide enthusiastically at first, roughly doubling in number with each division. But after about 40 to 60 divisions something strange happened. Cells would slow down and eventually stop dividing altogether, entering a state called senescence, kind of permanent retirement where cells remained alive but could no longer reproduce. We found this discovery, became known as Hayflick limit, was revolutionary, showed normal human cells have built-in counting mechanism, some cellular clock keeping track of how many times divided. After reaching limit simply stop as if run out of divisions. Direct evidence aging happens at cellular level, not just at whole organism level. Our cells themselves grow old. Each fact a gentle step in understanding slow emergence, the gradual realization that aging is not a simple process but a complex phenomenon woven into the very fabric of our biology. From ancient Greek theories about cooling and drying to Weismann's evolutionary insights, from cellular immortality questions to Hayflick's discovery of the division limit, humanity's slowly piecing together the puzzle. Feel yourself in that gradual understanding now, recognizing that your own cells carry this limit, this biological clock counting divisions, marking time. There's something peaceful in knowing this is natural, universal, written into life itself. There's nothing to solve tonight, nothing to resist. Just rest in the gentle acknowledgement that aging is part of being alive. A process as natural as breathing, as inevitable as the passage of time. Let yourself drift now with the rhythm of cell division, those millions of cells in your body dividing and renewing, each carrying its count, its limit, its eventual senescence, peaceful and natural, carried gently towards sleep. As we continue our peaceful journey through the story of aging, we move from the discovery that cells have a built-in division limit to understanding what causes this limit. The Hayflick limit raised a profound question. How do cells count their divisions? What biological mechanism acts as this cellular clock, keeping track of how many times a cell has replicated? The answer, when it came, revealed one of the most elegant molecular timepieces in all of biology. The key to understanding cellular aging turned out to be hiding at the very ends of our chromosomes. Chromosomes, as you may know, are the structures inside our cells that carry our genetic information in the form of DNA. Each chromosome is essentially a very long molecule of DNA wrapped around proteins. In the 1970s, scientists discovered that the ends of these chromosomes have special structures called telomeres, a word that comes from Greek roots meaning end parts. Telomeres are like the plastic tips at the ends of shoelaces, those little protective caps that keep the lace from fraying. In the same way, telomeres protect the ends of chromosomes from damage and from accidentally fusing with other chromosomes. But telomeres have another crucial property that connects them directly to aging. Every time a cell divides and copies its DNA, the telomeres get slightly shorter. A small piece of the telomere is lost with each cell division, like a candle burning down a little more each time it's lit. This discovery was made by several scientists working in the 1970s and 1980s. But perhaps the most important contributions came from Elizabeth Blackburn, Carol Greider, and Jack Szostak, who would eventually win the Nobel Prize in 2009 for their work on telomeres. They discovered that telomeres consist of repeating sequences of DNA. In humans, the sequence TTAGGG repeated thousands of times. These repetitive sequences don't code for any proteins. Their function is purely protective, like a buffer zone at the end of the chromosome. The reason telomeres shorten with each cell division has to do with a technical problem in DNA replication. When a cell copies its DNA, the molecular machinery that does the copying, an enzyme called DNA polymerase, can't quite reach the very end of the chromosome. It's a bit like painting yourself into a corner. The enzyme needs a starting point to begin copying, and at the very end of a linear chromosome, there's no room for that starting point. So with each replication, a small segment at the end remains uncopied, and the telomere gets shorter. This telomere shortening provides a natural explanation for the Hayflick limit. Each cell division shortens the telomeres a little more. After enough divisions, roughly 40 to 60 in human cells, the telomeres become critically short. When this happens, the cell recognizes that something is wrong. Short telomeres trigger a DNA damage response, and the cell stops dividing, entering that state of senescence that Hayflick had observed. The telomeres are acting as a molecular counting mechanism, a biological clock that tracks how many divisions a cell has gone through. This was a beautiful and satisfying explanation, but it immediately raised new questions. If telomere shortening causes cells to stop dividing, what about cells that need to keep dividing throughout our lives? Our bodies do contain some cells that continue dividing, stem cells in our bone marrow that produce new blood cells, stem cells in our intestines that renew the gut lining, and of course, the germ cells that become eggs and sperm. How do these cells avoid the telomere shortening problem? The answer lies in a remarkable enzyme called telomerase, which was discovered by Elizabeth Blackburn and Carol Greider in the mid-1980s. Telomerase is essentially a cellular fountain of youth. It can add DNA sequences back onto telomeres, rebuilding them and compensating for the shortening that occurs during DNA replication. Telomerase doesn't work in most adult cells, but it's active in stem cells, germ cells, and some other cells that need to maintain their division potential throughout life. The discovery of telomerase was thrilling because it showed that telomere shortening isn't absolutely inevitable. There's a biological mechanism that can counteract it. This raised an exciting possibility. Could activating telomerase in our cells prevent aging or even reverse it? The idea seemed almost too good to be true, and as scientists would discover, it was more complicated than it first appeared. There's a dark side to immortal cells. The problem is that cancer cells have also discovered the secret of telomerase. One of the hallmarks of cancer is uncontrolled cell division. Cancer cells divide again and again without stopping. Normal cells would hit the Hayflick limit and stop dividing, but cancer cells reactivate telomerase, rebuilding their telomeres and allowing them to divide indefinitely. In fact, about 85 to 90% of cancers have reactivated telomerase. This is how cancer cells achieve a kind of immortality, continuing to divide when they should have stopped long ago. This creates a difficult trade-off. Telomere shortening and cellular senescence act as a brake on cancer, preventing damaged cells from dividing endlessly. By limiting the number of times normal cells can divide, our bodies are protecting us from one of the deadliest threats we face. But this same protective mechanism contributes to aging, as more and more of our cells reach their Hayflick limit and become senescent. Our tissues lose their ability to regenerate and repair themselves. We're caught between the need to prevent cancer and the desire to maintain youthful tissues. And so we learned Hayflick limit raised profound question: how do cells count their divisions? What biological mechanism acts as this cellular clock, keeping track of how many times cell replicated? Answer: when it came, revealed one of biology's most elegant molecular timepieces, key to understanding cellular aging, turned out hiding at very ends of our chromosomes. We discovered chromosomes, structures inside cells carrying genetic information as DNA, are essentially very long DNA molecules wrapped around proteins. In 1970s, scientists discovered chromosome ends have special structures called telomeres, word from Greek roots meaning end parts. Telomeres like plastic tips at shoelace ends, little protective caps keeping lace from fraying. Same way telomeres protect chromosome ends from damage and from accidentally fusing with other chromosomes. We found telomeres have another crucial property connecting them directly to aging. Every time cell divides and copies DNA, telomeres get slightly shorter. Small telomere piece lost with each cell division, like candle burning down little more each time it's lit. Discovery made by several scientists working 1970s and 1980s, but perhaps most important contributions came from Elizabeth Blackburn, Carol Greider, and Jack Szostak, eventually won Nobel Prize 2009 for telomere work. Discovered telomeres consist of repeating DNA sequences. In humans, sequence TTAGGG repeated thousands of times. These repetitive sequences don't code for any proteins, function purely protective like buffer zone at chromosome end. We learned reason telomeres shorten with each cell division has to do with technical DNA replication problem. When cell copies DNA, the molecular machinery doing copying, enzyme called DNA polymerase, can't quite reach chromosome very end. Bit like painting yourself into corner, enzyme needs starting point to begin copying, and at linear chromosome very end there's no room for that starting point. So with each replication, small segment at end remains uncopied and telomere gets shorter. We discovered this telomere shortening provides natural Hayflick limit explanation. Each cell division shortens telomeres little more. After enough divisions, roughly 40 to 60 in human cells, telomeres become critically short. When this happens, cell recognizes something wrong. Short telomeres trigger DNA damage response and cell stops dividing, entering senescence state. Hayflick observed telomeres acting as molecular counting mechanism, biological clock tracking how many divisions cell gone through. We found beautiful and satisfying explanation, but immediately raised new questions. If telomere shortening causes cells to stop dividing, what about cells needing to keep dividing throughout our lives? Bodies do contain some cells continuing dividing, stem cells in bone marrow producing new blood cells, stem cells in intestines renewing gut lining, and of course germ cells becoming eggs and sperm. How do these cells avoid telomere shortening problem? We learned answer lies in remarkable enzyme called telomerase, discovered by Elizabeth Blackburn and Carol Greider mid-1980s. Telomerase, essentially cellular fountain of youth, can add DNA sequences back onto telomeres, rebuilding them and compensating for shortening occurring during DNA replication. Telomerase doesn't work in most adult cells but active in stem cells, germ cells, and some other cells needing to maintain division potential throughout life. We discovered telomerase discovery thrilling because showed telomere shortening isn't absolutely inevitable. There's biological mechanism that can counteract it. Raised exciting possibility. Could activating telomerase in our cells prevent aging or even reverse it? Idea seemed almost too good to be true, and as scientists would discover was more complicated than first appeared. There's dark side to immortal cells. We found problem as cancer cells also discovered telomerase secret. One of cancer hallmarks is uncontrolled cell division. Cancer cells divide again and again without stopping. Normal cells would hit Hayflick limit and stop dividing, but cancer cells reactivate telomerase, rebuilding their telomeres and allowing indefinite division. In fact, about 85 to 90% of cancers have reactivated telomerase. How cancer cells achieve kind of immortality, continuing to divide when should have stopped long ago. We learned this creates difficult trade-off. Telomere shortening and cellular senescence act as brake on cancer, preventing damaged cells from dividing endlessly. By limiting times normal cells can divide our bodies, protecting us from one of deadliest threats we face. But same protective mechanism contributes to aging. As more and more cells reach Hayflick limit and become senescent, our tissues lose ability to regenerate and repair themselves. Caught between need to prevent cancer and desire to maintain youthful tissues. Each fact a gentle step in understanding's deepening. The gradual realization that aging involves elegant molecular mechanisms, telomeres shortening like candles burning down, telomerase rebuilding them in special cells, the trade-off between cancer prevention and tissue renewal. From chromosome ends to DNA replication problems, from Nobel Prize discoveries to cancer's immortality trick, all revealing aging's cellular clockwork. Rest in this molecular understanding now, this recognition that your cells carry these telomeres, shortening with each division, counting down toward senescence. It's a protective mechanism as much as a limitation, keeping you safe from cancer even as it marks time's passage. Let yourself drift with these cellular processes, these telomeres shortening imperceptibly with each division, these occasional cancer cells held in check by the Hayflick limit, the trade-off between immortality and safety, peaceful and inevitable, carried gently towards sleep. As we continue our peaceful journey through the story of aging, we move beyond telomeres to explore the many other ways our bodies change over time. Cellular senescence and telomere shortening are important pieces of the aging puzzle, but they're not the whole picture. Aging is a multifaceted process involving damage accumulation, changes in how genes are expressed, the gradual decline of cellular repair systems, and the slow degradation of the molecular machinery that keeps us alive. Let's explore these gentle, inevitable changes. One of the most important concepts in understanding aging is the idea of accumulated damage. Throughout our lives, our cells and the molecules within them are constantly being damaged by both internal and external factors. Every time we breathe, every time our cells produce energy, harmful byproducts are created. The most important of these are called free radicals or reactive oxygen species, highly reactive molecules that contain oxygen and can damage virtually any biological molecule they encounter. Free radicals are produced continuously in our cells as a natural consequence of metabolism, particularly in the mitochondria, the tiny structures inside cells that generate energy. Mitochondria are sometimes called the powerhouses of the cell because they convert nutrients into a form of energy the cell can use, a molecule called ATP. But this energy production process is imperfect, and about 1 to 2% of the oxygen used by mitochondria is converted into free radicals rather than being fully processed. These free radicals can attack DNA, proteins, and lipids, the fatty molecules that make up cell membranes. This concept led to what's called the free radical theory of aging, proposed in the 1950s by Denham Harman, a chemist who wondered whether the damage caused by free radicals might accumulate over time and contribute to aging. Harman's idea was that aging is essentially the result of a slow accumulation of molecular damage, like rust gradually weakening a piece of metal, or repeated small nicks gradually dulling a knife blade. Over decades, the cumulative damage from free radicals might cause our cells and tissues to function less efficiently, leading to the decline we recognize as aging. The free radical theory was appealing because it explained why animals with higher metabolic rates, those whose cells burn energy faster, tend to have shorter lifespans. A mouse, for example, has a much higher metabolic rate than a human, and it lives only about 2 to 3 years. The theory suggested that the faster metabolism produces more free radicals, leading to faster damage accumulation and shorter life. It also explained why antioxidants, molecules that can neutralize free radicals, might be beneficial. Our cells produce their own antioxidants, like glutathione and superoxide dismutase, and we obtain others from our diet, like vitamins C and E. However, as appealing as the free radical theory was, it turned out to be an oversimplification. Research over the past few decades has shown that the relationship between free radicals and aging is more complicated than initially thought. Giving animals extra antioxidants, for instance, doesn't consistently extend their lifespan as the simple version of the theory would predict. And surprisingly, in some situations, free radicals seem to serve important signaling functions in cells rather than being purely harmful. Nevertheless, oxidative damage, damage caused by free radicals and related molecules, is certainly one of the factors that contributes to aging, even if it's not the whole story. Beyond free radical damage, another crucial aspect of aging involves what happens to proteins over time. Proteins are the workhorses of the cell. They carry out most of the functions necessary for life, from catalyzing chemical reactions to providing structural support to transporting molecules. But proteins don't last forever. They can be damaged, they can misfold, taking on incorrect three-dimensional shapes, and they can become modified by chemical reactions that impair their function. Young cells are quite good at dealing with damaged proteins. They have sophisticated quality control systems that can either repair damaged proteins or break them down and recycle their components. The most important of these systems is called the proteasome, a large molecular machine that acts like a cellular recycling center, breaking down damaged or unneeded proteins into their constituent amino acids, which can then be used to build new proteins. There's also another system called autophagy, meaning self-feeding, where cells can engulf and digest their own damaged components, including not just proteins but entire organelles like worn-out mitochondria. But as we age, these quality control systems become less efficient. The proteasome slows down, and autophagy becomes less active. Damaged proteins begin to accumulate, and some of them aggregate, clumping together into tangled masses that the cell can't easily clear away. In some cases, these protein aggregates become toxic, interfering with normal cellular functions. Many age-related diseases involve protein aggregation. Alzheimer's disease is characterized by aggregates of proteins called amyloid beta and tau in the brain. Parkinson's disease involves aggregates of a protein called alpha-synuclein, and many other neurodegenerative conditions show similar patterns. The accumulation of damaged proteins and the decline in cellular cleaning systems represents another example of how aging involves the gradual failure of maintenance mechanisms. When we're young, our cells can keep up with the constant wear and tear, repairing damage almost as quickly as it occurs. But over time, the repair systems themselves become damaged or less efficient, and damage begins to accumulate faster than it can be fixed. It's like a maintenance crew that's doing a good job keeping a building in repair, but gradually the crew gets smaller and less efficient, and eventually they can't keep up with all the needed repairs. Another major contributor to aging involves changes in our DNA that don't actually alter the genetic sequence itself, but rather change which genes are turned on or off. These are called epigenetic changes, modifications to DNA and the proteins that package it that affect gene expression without changing the underlying genetic code. Throughout our lives, our cells acquire epigenetic marks that can silence some genes and activate others, and these changes accumulate in ways that alter how cells function. One of the most studied epigenetic modifications is called DNA methylation, where small chemical groups called methyl groups are attached to certain positions in the DNA. The pattern of methylation across the genome changes predictably as we age, and these changes are so consistent that scientists have developed what they call epigenetic clocks, methods of estimating a person's biological age based on the methylation patterns in their DNA. These clocks can sometimes reveal that someone's cells are older or younger than their chronological age, suggesting that the rate of epigenetic aging varies from person to person. And so we learned one of most important aging understanding concepts is accumulated damage idea. Throughout lives our cells and molecules within them constantly being damaged by both internal and external factors. Every time we breathe, every time cells produce energy, harmful byproducts created, most important called free radicals or reactive oxygen species, highly reactive oxygen-containing molecules damaging virtually any biological molecule they encounter. We discovered free radicals produced continuously in cells as natural metabolism consequence, particularly in mitochondria, tiny cell structures generating energy, sometimes called cell powerhouses converting nutrients into usable energy form, ATP molecule. But energy production process imperfect, about 1 to 2% of mitochondria used oxygen converted into free radicals rather than being fully processed. Free radicals can attack DNA, proteins, and lipids, fatty molecules making up cell membranes. We found this concept led to free radical theory of aging proposed 1950s by Denham Harman, chemist wondering whether free radical damage might accumulate over time contributing to aging. Harman's idea, aging essentially result of slow molecular damage accumulation, like rust gradually weakening metal or repeated small nicks gradually dulling knife blade. Over decades, cumulative free radical damage might cause cells and tissues to function less efficiently, leading to decline we recognize as aging. We learned free radical theory appealing because it explained why animals with higher metabolic rates, cells burning energy faster, tend to have shorter lifespans. Mouse, for example, has much higher metabolic rate than human and lives only about 2 to 3 years. Theory suggested faster metabolism produces more free radicals, leading to faster damage accumulation and shorter life. Also explained why antioxidants, molecules neutralizing free radicals, might be beneficial. Cells produce own antioxidants like glutathione and superoxide dismutase, obtain others from diet like vitamin C and E. We discovered, however, appealing as free radical theory was turned out to be oversimplification. Research over past decades shown relationship between free radicals and aging more complicated than initially thought. Giving animals extra antioxidants, for instance, doesn't consistently extend lifespan as simple theory version would predict. Surprisingly, in some situations, free radicals seem to serve important cell signaling functions rather than being purely harmful. Nevertheless, oxidative damage caused by free radicals and related molecules, certainly one of factors contributing to aging, even if not whole story. We found beyond free radical damage, another crucial aging aspect involves what happens to proteins over time. Proteins are cell workhorses carrying out most life-necessary functions, from catalyzing chemical reactions to providing structural support to transporting molecules. But proteins don't last forever, can be damaged, can misfold, taking incorrect three-dimensional shapes, can become modified by chemical reactions impairing function. We learned young cells quite good at dealing with damaged proteins, have sophisticated quality control systems that can either repair damaged proteins or break them down and recycle components. Most important system called proteasome, large molecular machine acting like cellular recycling center, breaking down damaged or unneeded proteins into constituent amino acids which can then build new proteins. Also another system called autophagy, meaning cell feeding, where cells can engulf and digest own damaged components, including not just proteins but entire organelles like worn-out mitochondria. We discovered, but as we age, these quality control systems become less efficient, proteasome slows down, autophagy becomes less active. Damaged proteins begin accumulating in some aggregate, clumping together into tangled masses cell can't easily clear away. In some cases, protein aggregates become toxic, interfering with normal cellular functions. Many age-related diseases involve protein aggregation. Alzheimer's disease, characterized by amyloid beta and tau protein aggregates in brain. Parkinson's disease involves alpha-synuclein protein aggregates. Many other neurodegenerative conditions show similar patterns. We found damaged protein accumulation and cellular cleaning system decline represents another example of how aging involves gradual maintenance mechanism failure. When young, our cells can keep up with constant wear and tear, repairing damage almost as quickly as occurs. But over time, repair systems themselves become damaged or less efficient, and damage begins accumulating faster than can be fixed. Like maintenance crew doing good job keeping building in repair, but gradually crew gets smaller and less efficient, eventually can't keep up with all needed repairs. We learned another major aging contributor involves changes in DNA, not actually altering genetic sequence itself, but rather changing which genes turned on or off, called epigenetic changes. Modifications to DNA and proteins packaging it, affecting gene expression without changing underlying genetic code. Throughout lives, cells acquire epigenetic marks that can silence some genes and activate others. Changes accumulate in ways altering how cells function. We discovered one most studied epigenetic modifications called DNA methylation, where small chemical groups, methyl groups, attached to certain DNA positions. Methylation pattern across genome changes predictably as we age. Changes so consistent, scientists developed what they call epigenetic clocks, methods of estimating person's biological age based on DNA methylation patterns. Clocks can sometimes reveal someone's cells are older or younger than chronological age, suggesting epigenetic aging rate varies person to person. Each fact a gentle step in understanding's expansion, the gradual realization that aging involves multiple parallel processes, free radical damage accumulating, proteins misfolding and aggregating, cellular cleaning systems declining, epigenetic patterns shifting. From Harman's oxidative theory to proteasome recycling, from autophagy cell feeding to DNA methylation clocks, all contributing to time's passage. Rest in this multifaceted understanding now, this recognition that your cells face many challenges simultaneously: oxidative stress, protein damage, changing gene expression, all marking the years in different ways. It's not one thing but many, working together to create the aging process. Let yourself drift with these molecular changes, these free radicals occasionally escaping mitochondria, these proteins occasionally misfolding, these epigenetic marks slowly accumulating, peaceful and natural, the inevitable cost of living, carried gently towards sleep. As we continue our peaceful journey through the story of aging, we move from the molecular changes within cells to explore how aging affects entire organs and systems. The cumulative damage to DNA, proteins, and cellular machinery doesn't happen in isolation. It manifests in the visible signs of aging we all recognize. The graying of hair, the wrinkling of skin, the stiffening of joints, the gradual decline in strength and stamina. Each of these changes has its own biological story, its own connection to the fundamental aging processes we've been exploring. One of the most visible signs of aging is the graying of hair, a change so universal that it's become synonymous with growing older. Hair gets its color from pigment cells called melanocytes located in the hair follicles, the tiny pockets in the skin where each hair grows. These melanocytes produce melanin, the same pigment that colors our skin, and they inject this melanin into the growing hair shaft. Dark hair contains more melanin, particularly a type called eumelanin, while red hair contains a different type called pheomelanin, and blonde hair contains less melanin overall. As we age, the melanocytes in our hair follicles gradually lose their ability to produce melanin. This happens because the stem cells that replenish the melanocytes become depleted over time. Each hair goes through cycles of growth, rest, and shedding, and with each cycle, the melanocyte stem cell pool becomes smaller. Eventually, there aren't enough melanocytes left to pigment the hair, and new hair grows in white or gray, not actually gray in color but colorless, appearing gray because of the way the unpigmented hair mixes visually with still pigmented strands. The timing of graying is largely genetic, which is why some people go gray in their 20s while others maintain their hair color into their 60s or beyond. Another unmistakable sign of aging is the wrinkling and sagging of skin. This transformation reflects fundamental changes in the structure of the skin itself. Skin is composed of multiple layers, but the most important for understanding wrinkles are the outer epidermis and the deeper dermis. The dermis contains proteins called collagen and elastin that give skin its strength and elasticity. Collagen provides structural support, like the framework of a building, while elastin allows the skin to stretch and return to its original shape. Young skin contains abundant collagen and elastin arranged in well-organized networks. But with age, several things happen. First, the production of new collagen slows down. We make less and less as we get older. Second, existing collagen fibers become cross-linked and disorganized, making them stiffer and less functional. Third, elastin fibers become fragmented and lose their springiness. And fourth, external factors like sun exposure accelerate these changes through a process called photoaging, where ultraviolet radiation damages collagen and elastin directly. The result is skin that's thinner, less elastic, and prone to developing wrinkles and sagging. Beneath the skin, our bones also undergo significant age-related changes. Throughout our lives, bone is constantly being remodeled. Old bone tissue is broken down by cells called osteoclasts, and new bone is built up by cells called osteoblasts. In youth and early adulthood, these processes are balanced, maintaining bone density and strength. But as we age, particularly after middle age, the balance shifts. Bone breakdown begins to outpace bone formation, leading to a gradual loss of bone density. This bone loss affects everyone to some degree, but it's particularly pronounced in women after menopause because the hormone estrogen, which helps maintain bone density, drops dramatically. The resulting condition, osteoporosis, makes bones brittle and prone to fracture. The skeleton literally becomes more porous. The solid-looking bone develops more and larger spaces within it, like a sponge with increasingly large holes. This is why older adults are more susceptible to fractures from falls that might not have injured them when they were younger. The aging skeleton is another example of a maintenance system, the constant remodeling of bone, falling out of balance. Our cardiovascular system also changes with age in ways that affect our overall health and vitality. The heart itself undergoes subtle changes. The heart muscle can become slightly thicker and stiffer, making the heart work harder to pump the same amount of blood. The number of pacemaker cells, specialized cells in the heart that control its rhythm, declines, which can lead to irregular heartbeats. The heart valves can become thicker and stiffer, sometimes causing them to leak or not open fully. But perhaps more importantly, the blood vessels throughout the body undergo significant aging. The arteries, which carry oxygen-rich blood from the heart to the rest of the body, are normally elastic tubes that can expand with each heartbeat and then recoil. This elasticity helps maintain smooth blood flow and cushions the pulse. However, with age, arteries tend to stiffen, a condition sometimes called arteriosclerosis, literally hardening of the arteries. The walls of the arteries can become thicker and less flexible, partly due to the accumulation of calcium deposits and changes in the elastic fibers in the arterial walls. In addition to stiffening, arteries can also accumulate fatty deposits in their inner lining through a process called atherosclerosis. These deposits, called plaques, contain cholesterol, inflammatory cells, and other substances. They narrow the arteries, reducing blood flow, and they can rupture, causing blood clots that can lead to heart attacks or strokes. While atherosclerosis is influenced by many factors including diet, exercise, genetics, and smoking, age is one of the strongest risk factors. The longer we live, the more time these deposits have to accumulate. Our immune system, which protects us from infections and disease, also changes with age in a process called immunosenescence. Young immune systems are highly responsive, capable of mounting vigorous defenses against new pathogens and forming strong immunological memories that provide long-lasting protection after vaccination or infection. But aged immune systems become less effective in several ways. The thymus, a small organ in the chest where certain immune cells called T cells mature, begins to shrink after puberty and continues to atrophy with age. This means fewer new T cells are produced in older adults. The existing immune cells become less diverse. The repertoire of different immune cell types that can recognize different pathogens becomes more limited. Immune cells also become less responsive, mounting weaker responses to new infections and vaccinations. At the same time, aged immune systems often develop chronic low-grade inflammation, sometimes called inflammaging, a state where inflammatory signals are constantly present even without acute infection, contributing to various age-related diseases. And so we learned one of most visible aging signs is hair graying. Change so universal becomes synonymous with growing older. Hair gets color from pigment cells called melanocytes located in hair follicles, tiny skin pockets where each hair grows. Melanocytes produce melanin, same pigment coloring skin, inject melanin into growing hair shaft. Dark hair contains more melanin, particularly eumelanin type. Red hair contains different type, pheomelanin. Blonde hair contains less melanin overall. We discovered as we age, hair follicle melanocytes gradually lose ability to produce melanin. Happens because stem cells replenishing melanocytes become depleted over time. Each hair goes through growth, rest, shedding cycles. With each cycle, melanocyte stem cell pool becomes smaller. Eventually, not enough melanocytes left to pigment hair. New hair grows in white or gray, not actually gray in color but colorless, appearing gray because unpigmented hair mixes visually with still pigmented strands. Graying timing largely genetic, why some people go gray in 20s while others maintain hair color into 60s or beyond. We found another unmistakable aging sign is skin wrinkling and sagging. Transformation reflects fundamental skin structure changes. Skin composed of multiple layers, but most important for understanding wrinkles are outer epidermis and deeper dermis. Dermis contains proteins called collagen and elastin, giving skin strength and elasticity. Collagen provides structural support like building framework. Elastin allows skin to stretch and return to original shape. We learned young skin contains abundant collagen and elastin arranged in well-organized networks. But with age, several things happen. First, new collagen production slows down, make less and less as get older. Second, existing collagen fibers become cross-linked and disorganized, making them stiffer and less functional. Third, elastin fibers become fragmented and lose springiness. Fourth, external factors like sun exposure accelerate changes through process called photoaging. Ultraviolet radiation damages collagen and elastin directly. Result is skin that's thinner, less elastic, prone to developing wrinkles and sagging. We discovered beneath skin our bones also undergo significant age-related changes. Throughout lives, bone constantly being remodeled, old bone tissue broken down by cells called osteoclasts, new bone built up by cells called osteoblasts. In youth and early adulthood, processes balanced, maintaining bone density and strength. But as age, particularly after middle age, balance shifts. Bone breakdown begins outpacing bone formation, leading to gradual bone density loss. We found bone loss affects everyone to some degree, but particularly pronounced in women after menopause because hormone estrogen helps maintain bone density, drops dramatically. Resulting condition osteoporosis makes bones brittle and prone to fracture. Skeleton literally becomes more porous. Solid-looking bone develops more and larger spaces within, like sponge with increasingly large holes. Why older adults more susceptible to fractures from falls that might not have injured them when younger. Aging skeleton, another example of maintenance system. Constant bone remodeling, falling out of balance. We learned cardiovascular system also changes with age in ways affecting overall health and vitality. Heart itself undergoes subtle changes. Heart muscle can become slightly thicker and stiffer, making heart work harder to pump same blood amount. Number of pacemaker cells, specialized cells controlling heart rhythm, declines, can lead to irregular heartbeats. Heart valves can become thicker and stiffer, sometimes causing them to leak or not open fully. We discovered, but perhaps more importantly, blood vessels throughout body undergo significant aging. Arteries, carrying oxygen-rich blood from heart to rest of body, normally elastic tubes that can expand with each heartbeat then recoil. Elasticity helps maintain smooth blood flow and cushions pulse. However, with age, arteries tend to stiffen, condition sometimes called arteriosclerosis, literally hardening of arteries. Arterial walls can become thicker and less flexible, partly due to calcium deposit accumulation and elastic fiber changes in arterial walls. We found in addition to stiffening, arteries can also accumulate fatty deposits in inner lining through process called atherosclerosis. Deposits called plaques contain cholesterol, inflammatory cells, other substances. They narrow arteries, reducing blood flow, can rupture, causing blood clots leading to heart attacks or strokes. While atherosclerosis influenced by many factors, diet, exercise, genetics, smoking, age is one of strongest risk factors. Longer we live, more time these deposits have to accumulate. We learned immune system protecting us from infections and disease also changes with age and process called immunosenescence. Young immune systems highly responsive, mounting vigorous defenses against new pathogens, forming strong immunological memories, providing long-lasting vaccination or infection protection. But aged immune systems become less effective in several ways. We discovered thymus, small chest organ where certain immune cells called T cells mature, begins shrinking after puberty, continues atrophying with age, means fewer new T cells produced in older adults. Existing immune cells become less diverse, repertoire of different immune cell types recognizing different pathogens becomes more limited. Immune cells also become less responsive, mounting weaker responses to new infections and vaccinations. At same time, aged immune systems often develop chronic low-grade inflammation, sometimes called inflammaging, state where inflammatory signals constantly present even without acute infection, contributing to various age-related diseases. Each fact a gentle step in understanding's embodiment, the gradual realization that aging manifests throughout the body, in graying hair and wrinkling skin, in weakening bones and stiffening arteries, in the immune system's declining vigilance. From melanocyte depletion to collagen cross-linking, from bone remodeling imbalance to arterial stiffening, from thymic atrophy to inflammaging, all the visible signs of time passing. Rest in this bodily understanding now, this recognition that aging is not just cellular but systemic, affecting every organ and tissue. Your hair may gray, your skin may wrinkle, your bones may weaken. These are not failures but natural consequences of living, of time's patient work. Let yourself drift with these slow transformations, these melanocytes gradually exhausting, these collagen fibers slowly cross-linking, these bones imperceptibly thinning, these arteries progressively stiffening, peaceful and inevitable, the body's gradual settlement into age, carried gently towards sleep. As we continue our peaceful journey through the story of aging, we move from the visible physical changes to explore some of the deeper biological mysteries that researchers are still working to understand. While we know many of the mechanisms that contribute to aging, scientists continue to discover new pathways and processes, and perhaps most intriguingly, they're finding that aging rates can be influenced, that the biological clock can be sped up or slowed down by various factors. This brings us to questions about whether aging is truly inevitable and whether it might someday be slowed or even reversed. One of the most fascinating discoveries in aging research came from studies of caloric restriction, the practice of reducing food intake without malnutrition. Beginning in the 1930s, researchers found that laboratory animals fed significantly fewer calories than normal, typically about 30 to 40% less, lived substantially longer than animals allowed to eat freely. Rats on caloric restriction could live up to 50% longer than normal rats. This effect has been observed in yeast, worms, flies, fish, and many species of mammals, making it one of the most robust and reproducible findings in all of gerontology. The mechanism behind caloric restriction's life-extending effects is still being worked out, but it appears to involve several interconnected pathways. When the body senses that food is scarce, it shifts into a kind of survival mode, prioritizing maintenance and repair over growth and reproduction. Resources are redirected toward protecting cells and repairing damage rather than building new tissues. One of the key mediators of this response is a family of proteins called sirtuins, which become more active during caloric restriction and help cells respond to stress and maintain their function. Caloric restriction also affects metabolism in fundamental ways. It reduces the production of insulin and related hormones, which has widespread effects on how cells process nutrients and respond to growth signals. Lower insulin levels are associated with increased stress resistance and enhanced cellular cleanup processes like autophagy, the same self-eating mechanism we discussed earlier that clears away damaged components. In a sense, mild nutritional stress seems to keep cellular maintenance systems more active and efficient, fighting against the gradual decline that normally occurs with age. However, caloric restriction is not a simple solution to aging in humans. The degree of restriction needed to see lifespan benefits in laboratory animals is quite severe. Imagine eating 30 to 40% less than you currently do, every day, for your entire life. Most people would find this extremely difficult to sustain, and there are potential downsides including loss of bone density, muscle mass, and reproductive function. Long-term studies in primates have shown more modest benefits than seen in rodents, and human studies are still ongoing. Nevertheless, caloric restriction research has been invaluable for revealing biological pathways that influence aging, pathways that might be targeted by future interventions without requiring severe dietary restriction. Related to caloric restriction is the emerging field of research on fasting and time-restricted eating. Some studies suggest that it's not just the total amount of food consumed but also the timing and pattern of eating that matters. Intermittent fasting, alternating periods of eating and fasting, or time-restricted eating, confining all food intake to a limited window each day, may activate some of the same beneficial pathways as caloric restriction without requiring a permanent reduction in total calories. These eating patterns may give the body extended periods to complete repair processes and clear away cellular debris, though research in this area is still in relatively early stages. Another major influence on aging that researchers have identified is physical activity. Regular exercise, particularly as we age, has profound effects on maintaining function and extending health span, the period of life spent in good health, as opposed to lifespan, which is total years lived. Exercise benefits nearly every system in the body. It maintains muscle mass and strength, helping to prevent the age-related muscle loss called sarcopenia. It strengthens bones, reducing osteoporosis risk. It improves cardiovascular function, keeping arteries more flexible and the heart stronger. At the cellular level, exercise promotes the creation of new mitochondria, those energy-generating structures we discussed earlier, through a process called mitochondrial biogenesis. This is particularly important because mitochondria themselves accumulate damage with age, becoming less efficient in producing more free radicals. By stimulating the production of fresh, healthy mitochondria, exercise helps maintain cellular energy production and reduce oxidative stress. Exercise also promotes autophagy, enhancing the cell's ability to clean up damaged components. In the brain, exercise has particularly striking effects. It stimulates the production of growth factors that support the health and growth of neurons, and it promotes neurogenesis, the birth of new neurons, particularly in a region called the hippocampus that's crucial for memory formation. Regular physical activity is one of the most effective known ways to reduce the risk of age-related cognitive decline and dementia. People who exercise regularly throughout their lives maintain better cognitive function into old age than sedentary individuals. Beyond diet and exercise, genetics play a significant role in determining how fast we age and how long we live. Family longevity studies have shown that if your parents and grandparents live to advanced ages, you're more likely to do so as well, suggesting inherited factors influence aging. Researchers have identified numerous genes that affect lifespan in various organisms, from yeast to worms to flies to mice. Some of these genes are involved in stress resistance, DNA repair, or cellular maintenance processes we've already discussed. One of the most studied longevity genes is called FOXO3, which is more common in centenarians, people who live to 100 or beyond, than in the general population. FOXO3 helps regulate the expression of genes involved in stress resistance and DNA repair. Other longevity-associated genes affect growth hormone and insulin signaling, inflammation, or cholesterol metabolism. However, in humans, aging and lifespan are influenced by many genes, each contributing a small effect, rather than by any single longevity gene. The genetic contribution to lifespan is estimated to be about 20 to 30%, meaning that lifestyle and environmental factors account for the majority of variation in how long people live and how healthy they remain as they age. The discovery that aging can be influenced by diet, exercise, and genes has led researchers to wonder whether aging might be more malleable than previously thought. If cellular maintenance systems can be kept active through caloric restriction or exercise, if genes that promote longevity can be identified, might it be possible to develop interventions that slow aging directly? This has led to an active search for what are sometimes called geroprotectors, compounds that protect against aging. And so we learned one of most fascinating aging research discoveries came from caloric restriction studies, practice of reducing food intake without malnutrition. Beginning 1930s, researchers found laboratory animals fed significantly fewer calories than normal, typically about 30 to 40% less, lived substantially longer than animals allowed to eat freely. Rats on caloric restriction could live up to 50% longer than normal rats, effect observed in yeast, worms, flies, fish, many mammal species, making it one of gerontology's most robust and reproducible findings. We discovered mechanism behind caloric restriction's life-extending effects still being worked out, but appears involving several interconnected pathways. When body senses food scarcity, shifts into kind of survival mode, prioritizing maintenance and repair over growth and reproduction. Resources redirected toward protecting cells and repairing damage rather than building new tissues, One key mediator is sirtuin protein family becoming more active during caloric restriction, helping cells respond to stress and maintain function. We found caloric restriction also affects metabolism in fundamental ways, reduces insulin and related hormone production, having widespread effects on how cells process nutrients and respond to growth signals. Lower insulin levels associated with increased stress resistance and enhanced cellular cleanup processes like autophagy, same cell-feeding mechanism clearing away damaged components, and since mild nutritional stress seems to keep cellular maintenance systems more active and efficient, fighting against gradual decline normally occurring with age. We learned, however, caloric restriction not simple human aging solution. Degree of restriction needed to see laboratory animal lifespan benefits quite severe. Imagine eating 30 to 40% less than currently do every day for entire life. Most people would find extremely difficult to sustain. Potential downsides including bone density loss, muscle mass loss, reproductive function loss. Long-term primate studies shown more modest benefits than seen in rodents, human studies still ongoing. Nevertheless, caloric restriction research invaluable for revealing biological pathways influencing aging, pathways that might be targeted by future interventions without requiring severe dietary restriction. We discovered related to caloric restriction is emerging fasting and time-restricted eating research field. Some studies suggest not just total food amount consumed but also eating timing and pattern matters. Intermittent fasting, alternating eating in fasting periods, or time-restricted eating, confining all food intake to limited daily window, may activate some same beneficial pathways as caloric restriction without requiring permanent total calorie reduction. Eating patterns may give body extended periods to complete repair processes and clear cellular debris, though research in areas still relatively early stages. We found another major aging influence researchers identified as physical activity. Regular exercise, particularly as we age, has profound effects maintaining function and extending health span. Period of life spent in good health as opposed to lifespan, which is total years lived. Exercise benefits nearly every body system, maintains muscle mass and strength, helping prevent age-related muscle loss called sarcopenia, strengthens bones reducing osteoporosis risk, improves cardiovascular function keeping arteries more flexible and heart stronger. We learned at cellular level exercise promotes new mitochondria creation, those energy-generating structures, through process called mitochondrial biogenesis, particularly important because mitochondria themselves accumulate damage with age, becoming less efficient and producing more free radicals. By stimulating fresh, healthy mitochondria production, exercise helps maintain cellular energy production and reduce oxidative stress. Exercise also promotes autophagy, enhancing cells' ability to clean up damaged components. We discovered in brain exercise has particularly striking effects, stimulates growth factor production supporting neuron health and growth, promotes neurogenesis, new neuron birth, particularly in hippocampus region crucial for memory formation. Regular physical activity, one of most effective known ways to reduce age-related cognitive decline and dementia risk. People exercising regularly throughout lives maintain better cognitive function into old age than sedentary individuals. We found beyond diet and exercise, genetics play significant role determining how fast we age and how long we live. Family longevity studies shown if parents and grandparents live to advanced ages, you're more likely to do so, suggesting inherited factors influence aging. Researchers identified numerous genes affecting lifespan in various organisms, Yeast to worms to flies to mice, some genes involved in stress resistance, DNA repair, or cellular maintenance processes already discussed. We learned one most studied longevity genes called FOXO3 more common in centenarians, people living to 100 or beyond, than in general population. FOXO3 helps regulate gene expression involved in stress resistance and DNA repair. Other longevity-associated genes affect growth hormone and insulin signaling, inflammation, or cholesterol metabolism. However, in humans, aging and lifespan influenced by many genes, each contributing small effect rather than any single longevity gene. Genetic contribution to lifespan estimated about 20 to 30%, meaning lifestyle and environmental factors account for majority of variation in how long people live and how healthy they remain as age. We discovered that aging can be influenced by diet, exercise, and genes led researchers to wonder whether aging might be more malleable than previously thought. If cellular maintenance systems can be kept active through caloric restriction or exercise, if longevity-promoting genes can be identified, might be possible to develop interventions slowing aging directly? Led to active search for what's sometimes called geroprotectors, compounds protecting against aging. Each fact a gentle step in understanding's hopefulness. The gradual realization that aging is not entirely fixed but can be influenced by what we eat, how we move, what genes we inherit. From caloric restrictions survival mode to sirtuin stress response, from intermittent fasting to exercise-induced mitochondrial biogenesis, from FOXO3 longevity genes to geroprotector searches, all suggesting aging's malleability. Rest in this nuanced understanding now, this recognition that while aging is inevitable, some control exists over its pace. Diet matters, exercise matters, genes matter, but lifestyle accounts for most of the variation. You're not entirely at fate's mercy. Let yourself drift with these possibilities, these cellular maintenance systems responding to nutritional signals, these mitochondria multiplying with exercise, these longevity genes expressing themselves, peaceful and empowering, the knowledge that choices matter, carried gently towards sleep. As we approach the end of our peaceful journey through the story of aging, we turn to the frontiers of aging research and the profound questions that remain. Scientists have made remarkable progress in understanding why we age, but many mysteries persist, and the ultimate question, whether aging can be slowed, stopped, or even reversed, remains open. The search for interventions that might extend healthy lifespan continues, bringing together biology, medicine, and technology in ways that would have seemed like science fiction just decades ago. One of the most promising areas of current aging research involves a class of drugs called senolytics, compounds designed to selectively eliminate senescent cells from the body. Remember those cells that reach the Hayflick limit and stop dividing? They don't just sit quietly. Senescent cells secrete a mixture of inflammatory molecules, growth factors, and enzymes that can damage surrounding tissues, a phenomenon called the senescent-associated secretory phenotype, or SASP. As we age, senescent cells accumulate in our tissues, and their inflammatory secretions contribute to many age-related problems including arthritis, atherosclerosis, and tissue dysfunction. The idea behind senolytic drugs is simple but elegant. If senescent cells are causing problems, what if we could selectively remove them? In experiments with mice, researchers have used various senolytic compounds to clear senescent cells from aged animals. The results have been striking. Treated mice show improvements in numerous age-related conditions. Their fur becomes thicker and shinier, their kidneys function better, their hearts are healthier, and they even live longer. Some senescent cell clearance experiments have extended mouse health span by the equivalent of many human years. Several senolytic compounds are now being tested in human clinical trials for specific age-related diseases. These drugs work by targeting survival pathways that senescent cells rely on to resist their own death. Different senolytics target different pathways, and combinations of drugs may be needed to clear senescent cells from different tissues. While it's too early to know whether senolytics will prove safe and effective for human health span extension, the concept represents a fundamentally new approach to aging, treating it not by trying to prevent all damage but by periodically clearing away damaged cells that have accumulated. Another cutting-edge area of research involves cellular reprogramming, the possibility of reversing cellular age by changing gene expression patterns. This idea emerged from the discovery that mature, specialized cells can be reprogrammed back to a stem cell-like state. In 2006, Japanese researcher Shinya Yamanaka discovered that by activating just 4 specific genes, now called Yamanaka factors, adult cells could be converted into induced pluripotent stem cells, or iPSCs. These reprogrammed cells behave like embryonic stem cells, capable of becoming any cell type in the body. Yamanaka won the Nobel Prize for this breakthrough in 2012. What makes this relevant to aging is that the reprogramming process appears to erase many of the molecular signatures of aging. The reprogrammed cells have longer telomeres, fewer DNA methylation marks associated with age, and restored cellular function. This raises a tantalizing possibility. Could partial reprogramming, activating the Yamanaka factors briefly, not enough to fully dedifferentiate cells but enough to reset some aging markers, rejuvenate old cells without losing their specialized identity? Early experiments have shown promising results. In 2016, researchers used partial reprogramming to extend the lifespan of mice with progeria, a genetic condition that causes rapid aging. More recently, studies have shown that brief activation of Yamanaka factors can restore youthful function to aged tissues in normal mice, improving vision in old mice by rejuvenating retinal cells, for instance. The challenge is finding the right balance, enough reprogramming to reverse aging changes, but not so much that cells lose their identity or become cancerous. This is an area of intense current research with profound implications for the future of aging intervention. Beyond senolytics and reprogramming, scientists are exploring numerous other potential geroprotectors. Metformin, a widely used diabetes drug, has shown promise in extending lifespan and health span in various organisms and is being tested in large human trials for its anti-aging effects. Rapamycin, an immunosuppressant drug that inhibits a protein called mTOR, extends lifespan in mice and other organisms, though its side effects make long-term use in healthy people problematic. NAD+ boosters, compounds that increase levels of a molecule called nicotinamide adenine dinucleotide that declines with age, are being investigated for their potential to improve mitochondrial function and cellular energy metabolism. Resveratrol, a compound found in red wine and grapes, received significant attention for its potential to activate sirtuins and mimic some effects of caloric restriction, though subsequent research has been mixed on its effectiveness. The field is full of candidate compounds, each targeting different aspects of aging biology: inflammation, oxidative stress, protein quality control, mitochondrial function, stem cell exhaustion. The challenge is determining which interventions are truly effective, safe for long-term use, and able to extend healthy human lifespan rather than just showing effects in laboratory animals. As research continues, a fundamental question remains: is there a limit to human lifespan? Throughout most of human history, life expectancy was quite short, perhaps 25 to 40 years on average in ancient and medieval times, with many people dying from infections, accidents, or childbirth. But this doesn't reflect how long humans could live if they survived these hazards. Even in ancient times, some people lived to 70, 80, or occasionally beyond. What has changed dramatically over the past century and a half is the number of people who survive to old age. In 1900, life expectancy at birth in developed countries was about 45 to 50 years. Today it's around 80 years, an increase of about 30 years in just over a century. But this remarkable achievement has come primarily from reducing deaths at younger ages through better nutrition, sanitation, antibiotics, vaccines, and medical care. We've gotten much better at helping people reach old age, but the maximum human lifespan hasn't changed much. The oldest documented person ever, Jean Calment of France, lived to 122 years. While life expectancy has soared, the maximum lifespan seems stubbornly resistant to change. Some researchers believe there may be a biological limit to human lifespan, perhaps around 120 to 125 years, beyond which the accumulated damage and failure of multiple systems makes survival impossible. Others argue that if we can address the underlying processes of aging, clearing senescent cells, repairing DNA damage, maintaining stem cell function, preventing protein aggregation, there may be no fixed limit, and lifespans could potentially extend far beyond current maxima. The debate continues, but most researchers agree that even if maximum lifespan can't be greatly extended, health span, the years spent in good health, certainly can be. This brings us to perhaps the most important point about aging research. The goal is not just to live longer but to live better for longer. Extending lifespan without addressing the disabilities and diseases of old age would simply mean more years of frailty and suffering. But interventions that target the fundamental processes of aging hold the promise of compressing morbidity, keeping people healthier for more of their lives, so that illness and disability are confined to a brief period at the end of life rather than stretching across decades. And so our journey through the science of aging comes to its end, though the research continues, moving forward with new discoveries each year. We've traveled from ancient Greek theories about cooling and drying, through the discovery of cells and the realization that cells themselves age, to the molecular clockwork of telomeres and the cellular counting mechanism of the Hayflick limit. We've learned that aging involves accumulated damage, from free radicals attacking molecules, from proteins misfolding and aggregating, from DNA acquiring mutations and epigenetic changes. We've seen how this damage manifests throughout the body, in graying hair and wrinkling skin, in weakening bones and stiffening arteries, in the immune system's declining vigilance. We've discovered that aging can be influenced by our choices, that caloric restriction, exercise, and lifestyle factors account for the majority of variation in how we age. That genes play a role but not a dominant one. that cellular maintenance systems can be kept more active through the right interventions, whether dietary, physical, or potentially pharmaceutical. We've explored the frontiers of aging research, senolytic drugs to clear damaged cells, cellular reprogramming to reverse aging markers, various geroprotective compounds targeting different aging processes. We've contemplated whether there are limits to human lifespan and recognized that the true goal is not just longevity but health span, years lived in vitality and good health. As you rest now, know that you carry all of this within you. Your cells are dividing and senescing, your telomeres are shortening, your proteins are occasionally misfolding, your mitochondria are producing free radicals. But your cells are also repairing damage, clearing away debris, maintaining the intricate balance that keeps you alive and healthy. Aging is not a failure, it's the inevitable consequence of being alive, of experiencing the passage of time. There's profound peace in accepting aging as natural while knowing that the pace can be influenced. You age a little more each day, as does every living thing, but how you age is not entirely predetermined. The choices you make, how you eat, whether you move, how you engage with the world, all contribute to the quality of your aging. Rest in this understanding now. Your body is aging at its own pace, neither too fast nor too slow, following the biological programs written into your cells billions of years ago by evolution. Telomeres shorten. Proteins misfold. Cells senesce. But this happens slowly, patiently, over decades. Tonight, this moment, you are exactly the age you are, no younger, no older, perfectly positioned in time. Let yourself drift now with the gentle rhythm of aging, these processes unfolding imperceptibly day by day, year by year. Damage accumulating but being repaired, maintenance systems working tirelessly, the body gradually changing but still maintaining its essential functions. There's no rush, no urgency. Time passes at its own pace. Sleep now, knowing that aging is not something to fear but to understand. It's the price of living, the cost of all the years you've experienced, all the moments you've witnessed. Your cells have worked tirelessly to keep you alive, to maintain you, to repair you. They'll continue this work while you sleep, as they have every night of your life. Tomorrow you'll be very slightly older than you are today, your telomeres very slightly shorter, a few more senescent cells in your tissues, a few more epigenetic marks on your DNA. But you'll also have another day of life, another opportunity to influence how you age, another chance to appreciate the remarkable biological machinery that is your body. Good night, aging exactly as you should, moment by moment, cell by cell, year by year. Good night to the young cells still dividing, to the senescent cells that have earned their rest, to the proteins folding and unfolding, to the mitochondria producing energy and occasional free radicals. Rest now in the gentle inevitability of time's passage. You are aging, and that's okay. That's beautiful. That's life.